So I'm talking to Ian Henderson. Thanks for joining me, Ian. We're going to ask a few questions for the Arabidopsis Research Roundup this week. And Ian's going to talk about his new paper published in Genes Development with the title DNA Methylation Epigenetically Silences Crossover Hotspots and Controls Chromosomal Domains of Meiotic Recombination in Arabidopsis. Catchy title there. Ian, catchy, that's right. All right, so can I ask you to summarise the paper first of all, Ian?
Sure. So the paper's looking at recombination that happens between chromosomes, and specifically during the cell division called meiosis, which is it's a specialized cell division that creates gametes. So these are the sex cells. It's the same cell division that happens when humans reproduce, and plants do things in a very similar way at that sort of basic level. And then specifically what we're interested in are these regions called hotspots. And so these are narrow regions of the chromosome that have a much higher chance of recombination than other parts. And so a sort of general question in the lab is what controls those hotspots? And one of the hypotheses we're testing is that epigenetic information is important. And so in this paper, one of the things we do is take one of these epigenetic marks, DNA methylation, and add those to one of these hotspots. And what we show is that basically turns off the recombination. And so you could have two hotspots with the same sequence at the DNA level, but they have this different epigenetic mark. The DNA has become methylated, and that completely changes what's happening in terms of the recombination. And so that's one of the major parts of the paper. We also do some more work in there as well, looking at how DNA methylation and recombination play out at much broader scales right along the chromosomes.
Okay.
That's it.
Yeah, that sounds good. So can you let us know about what are the major experimental techniques that you've used in the paper, and is there any novelty in what you've done with those?
So one of the main way we measure recombination is to mark the chromosome somehow. So we might use a genetic difference, like a base pair difference between two chromosomes, or a gene making something very obvious, like a fluorescent protein of different colours. And so what we do is then follow those differences through meiosis and see whether these different variants get mixed up or not. And so that's the basic way that we measure recombination. The thing that we combine that with here is a trick that I learned as a postdoc in Steve Jacobson's lab. And basically it's a way that you can use small RNAs to direct DNA methylation to specific sequences. And so because we'd done that before, we decided to use that. So once we found where some hotspots were, we used that technique to basically add the methyl group specifically to the hotspots, and that's the sort of main.
Okay. Yeah. So that work with the siRNAs, of course, was featured in a Arabidopsis Research Roundup a couple of weeks ago. Some work that you did with Steve Jacobson published in Cell as well. So that was pretty.
My contribution, the experiment that I did that went into that paper, I did probably like 10 years ago.
Right. Okay.
But it was some northern blots that I did at that time, looking at different Dyson mutations. And they just never got published. They were sort of in the back of one of my lab books.
Yeah, okay. It's not a bad way to have them come out in a Cell paper. Yeah, that's right.
I was very pleased when Steve got in touch. We had to track them down, which was a bit of work.
Yeah. That's all right. Well worth it, though. So final question about the current paper. So the discoveries that you found and published in this paper, has it led you on to ask further questions on this topic?
It has, and the silencing of the hotspots in some way was what we expected. So the part that's still slightly surprising to us is what happened at the chromosome scale when we removed methylation, and it was somewhat unexpected, and so specifically because when we add the methylation to the hotspot, recombination went down, but then when we removed methylation, particularly from the centromeric regions, some of those regions actually went up in recombination. So it's the opposite result than we expected. And so we've been very surprised by this for a long time. But what it's led us to do is look more closely at the thing about heterochromatin, or these silent parts of the chromosomes, is DNA methylation is just one modification that's there. There's also lots of other ones that are interacting. And what we're looking at now is, well, if we change other epigenetic marks in these regions, maybe that will then give us the expected result. And we already have some preliminary data.
Okay.
That's quite interesting.
So you're going to be using different histone modifications and the
exactly,
like, things like that.
Exactly.
All right. Excellent. Yeah. Well, we'll look out for that work in the future. All right. Thanks very much, Ian, for explaining this paper for us. Cheers.
Okay. Thank you.